as I have told you that this is a 5-step process. So first I will explain you the first step in full detail. So for example, here we have a cell, we have adipocyte, and in the adipocyte what we have, triacylglycerol. This triacylglycerol is converted into diacylglycerol, and the enzyme involved here is adipocyte triglyceride lipase enzyme, which converts this triacylglycerol into diacylglycerol, which means one fatty acid is now removed from the triacylglycerol. If you remember the structure of triacylglycerol, there is a one molecule of glycerol which is a 3-carbon compound, and 3 fatty acids, they are attached to that glycerol. So that's why we name it as triacylglycerol. And with the help of this enzyme, one molecule of fatty acid is removed from the triacylglycerol, and we have left now that is the diacylglycerol. This is most important compound, and this diacylglycerol is also involved in signal transduction, and this diacylglycerol is further converted into monoacylglycerol with the help of enzyme which we name as hormone sensitive lipase. This is rate limiting enzyme. This is, we can say that, reaction controlling enzyme, which is also abbreviated as HSL, hormone sensitive lipase. And I will explain its regulation of this hormone sensitive lipase, that how this hormone sensitive lipase is involved in the regulation of this step, in the regulation of lipolysis. And then from the monoacylglycerol, it is converted into free fatty acids plus glycerol, and enzyme involved is again monoacylglycerol lipase. So first of all, let's see the regulation of this HSL. Regulation of HSL. This enzyme actually this works like glycogen phosphorylase enzyme. Like it has two forms. This enzyme can be in the form of phosphorylated and it can be in the form of dephosphorylated. Whenever this is in the form of phosphorylated, this enzyme is in the form of phosphorylated, this is active, and whenever it is in the form of dephosphorylated form, it is inactive. Question is, what kind of hormones which are involved in the activation of this hormone and inactivation of this hormone, in activation of this enzyme and inactivation of this enzyme? So let me explain it with the diagrammatic representation. For example, here we have a cell membrane. This is the cell membrane, suppose. And we know very well that in the cell membrane we have receptors. In the hormones which are involved, those are epinephrine as well as glucagon or growth hormones or thyroid hormones which promote the lipolysis. Hormones, such type of the hormones which promote the lipolysis, which are involved in the breakdown of fatty acids, in the breakdown of triacylglycerols, like growth hormone, thyroid hormone or epinephrine. When they become attached to this point, to this receptor, and usually the receptor involved is beta adrenergic receptor. What type of this receptor is? This receptor is beta adrenergic receptor. And after being attached to this receptor, when this receptor will become active, it will activate another receptor molecule which is usually named as adenylate cyclase. So it is going to activate this adenylate cyclase. After the activation of this adenylate cyclase, there will be the formation of cyclic AMP. This cyclic AMP will activate another enzyme which is usually called as protein kinase A. When this protein kinase A will become active, it will phosphorylate HSL. It will phosphorylate this is suppose HSL, and it will phosphorylate this HSL, and HSL, we can say that this HSL will become active. When this HSL will become active, it will start lipolysis. It will start lipolysis. There is another function of this signal cascade, that is the activation of perilipin protein. Usually the fat droplets, these fat droplets, these are covered with the help of perilipin. We have for example fat droplets, these are covered with another protein which is named as perilipin. These fat droplets are coated with this protein which is the perilipin. This pKa, this will, when phosphorylate this perilipin, then these fat droplets, they will become available to this hormone sensitive lipase like this. As a result of the phosphorylation of perilipin, of this perilipin, fat droplets, they will become exposed fat droplets, they will become exposed. Now HSL, which is active, now this can attack these fat droplets and start the lipolysis. Once again I am going to repeat that in the adipocytes we have triacylglycerol molecules, and first of all adipocyte triglyceride lipase, with the help of this enzyme, triacylglycerols are converted into diacylglycerol. Diacylglycerol is it also acts as a signaling molecule. Diacylglycerol, it will be further converted into monoacylglycerol with the help of another enzyme which is hormone sensitive lipase, and this is the most important enzyme and rate limiting enzyme. After being converted into monoacylglycerol, this monoacylglycerol is converted into free fatty acids and glycerol with the help of monoacylglycerol lipase enzyme. Is that clear? Now, what is the significance of this hormone sensitive lipase? This hormone sensitive lipase is very significant because it is controlled with the help of hormones. And how it is controlled or regulated with the help of hormones? This HSL, it actually exists in two forms, either in the form of phosphorylated form or in dephosphorylated form. Whenever it is in the phosphorylated form, it is active, and whenever it is in the dephosphorylated form, it is inactive. Now the question comes into the mind, what are those hormones which are going to activate it or which are going to phosphorylate this hormone sensitive lipase? Listen to me. What kind of hormones which are going to activate this hormone sensitive lipase, or what are the hormones which are going to phosphorylate this hormone sensitive lipase? And those hormones are epinephrine, growth hormone, thyroid hormones, or glucagon, which these are the hormones which are used whenever we are deficient with energy, whenever we are deficient with glucose, or whenever we are on starvation. All such type of the hormones which are used for the increased metabolic rate or which are produced during the starvation phase. Those hormones, through beta adrenergic receptors, those hormones will activate the adenylate cyclase, and as a result of this activation there will be the activation of cyclic AMP, and the cyclic AMP, it will activate the pKa. Now pKa has two functions. Number one, first it will phosphorylate this HSL, and when this HSL is phosphorylated, it will become active, and when it will become active, it will start lipolysis. And the second function of pKa is to phosphorylate the paralipin. What is it? Paralipin is a coat on the fat droplet, and when this paralipin is phosphorylated, now this fat droplet is exposed to HSL. It is exposed to HSL. Now HSL can attack on this fat droplet, and it can form the free fatty acids and glycerol, or in other words you can say that it will after attacking this fat droplet, it will start the lipolysis, and as a result of that lipolysis, the end products will be the fatty acids plus glycerol. So this was the first step in the oxidation of fatty acids. I hope you are clear with this. And second thing about the regulation, about the dephosphorylation, the insulin hormone. Which hormone? Insulin hormone, it is responsible for the deactivation or dephosphorylation of this hormone sensitive lipase through the help of phosphatase protein, through the help of enzyme phosphatase. It will convert phosphorylated hormone sensitive lipase into dephosphorylated hormone sensitive lipase, and when it is converted into dephosphorylated form, it will become inactive. Is that clear? So these epinephrine, growth hormone, these will activate this hormone sensitive lipase or phosphorylate this hormone sensitive lipase through cyclic AMP cascade, and there pK, it will function in two ways. One, the phosphorylation of HSL. Number two, the phosphorylation of paralipin. What will be the result of these two phosphorylations? The result will be the activation of hormone sensitive lipase and the exposure of this fat droplet to this active enzyme, and as a result the lipolysis will result. And whenever this enzyme has to be inactivate, at that time insulin hormone will come. It will activate the phosphatase enzyme, and phosphatase enzyme simply will remove the phosphate from this enzyme HSL, and it is again now in dephosphorylated form. When it is in dephosphorylated form, it will become inactive and lipolysis will stop. So this was the, we can say, the first step of oxidation of fatty acid.